Hello, my name is Clinton Gabel, and I am a third-year Ph.D. candidate in the Lei-Fu Chang Research Group at Purdue University. Today I'll be giving my talk, Anti-CRISPRs, the ongoing battle between phage and their foes. So first I'd like to talk about the type 1 CRISPR-Cas system found in Pseudomonas aeruginosa. As part of this system, there is the CSY complex. The CSY complex is seen here. It is created by one copy of Cas6f, 6 copies of Cas7.4f, 1 copy of Cas5.4f, and 1 copy Cas8.4f. And it is all integrated by one single CRISPR RNA. Whenever this CSY complex or surveillance complex finds a piece of double-stranded DNA that it matches with, it hybridizes with it. When it hybridizes, it causes a rotation of this Cas8.4f. And this rotation of Cas8.4f then attracts a Cas2,3 nuclease to degrade the matching double-stranded DNA. This matching double-stranded DNA is often from a mobile genetic element or invading phage. Therefore, this is a good mechanism for the Pseudomonas bacteria to invade infection. However, in response, mobile genetic elements or phages have started to encode anti-CRISPR proteins. These anti-CRISPR proteins often function by either inhibiting the surveillance complex or the nuclease, therefore shutting down this particular CRISPR system that has been inhibiting them. So in this particular study, we studied 3 different inhibitors. The first inhibitor that we studied was I-F7. Over here on the right, we have a SEC profile and SDS page, and we took this particular aliquot and did cryo-EM. And when we did cryo-EM, we determined the structure over here on the left. And when we determined the structure, we discovered that I-F7 behaves as a double-stranded DNA mimic. So to test this, we did an electromobility shift assay, and that is exactly what we showed here. Without the inhibitor, CSY can bind double-stranded DNA. However, with increasing concentrations of I-F7, double-stranded DNA binding is blocked. The next inhibitor that we studied was the IF14. Over here on the right, we once again have an SEC profile with SDS page. When we did cryo-EM, we noticed that there was a 2-to-1 stoichiometry of 2 copies of IF14 binding to 1 copy of CSY. Its method of action is seen to occlude the CRISPR RNA, therefore preventing double-stranded DNA CRISPR RNA hybridization. And to test this, we once again did an EMSA assay. However, there does seem to be a little bit of residual double-stranded DNA binding, which we attribute to a lower resolution helical bundle at the N-terminus of the different AcrIF14 subunits. So this is similar to other AcrIF inhibitors that seem to have nonspecific double-stranded DNA binding. The next inhibitor that we studied was AcrIF4. Once again, over on the right, we have the SEC profile and the SDS page. However, when we did cryo-EM, we noticed that this particular inhibitor seemed to have a structure different than that of the other AcrIF inhibitors. For instance, it seems to interact with the 4-helix bundle of Cas8.4 necessary for Cas2,3 recruitment. So if we look closer at this particular structure, we can see this hook at the top. And this hook actually will bind to this helical bundle and therefore lock the CSY complex in an inactive conformation. You can see that here in this particular animation. Over on the right in cyan is the inhibitor, AcrIF4, and in blue you can actually see the movement that is necessary for Cas2,3 recruitment. So this particular movement and this particular binding site of AcrIF4 should have no effect on double-stranded DNA binding. And so when we tested, again, using EMSA to see if there was any effect on double-stranded DNA binding, we didn't observe any. So one thing that we noticed while we were doing this particular study is that there appeared to be some convergent evolution among these AcrIF inhibitors. AcrIF2, 7, and 10 all appeared to be double-stranded DNA mimics, while AcrIF1, 9, and 14 all seem to bind to the same sites on the Cas7.4f backbone, preventing double-stranded DNA CRISPR-RNA hybridization. So I'd like to acknowledge the members of my lab and those who helped us at the cryo-EM facility to do this work, as well as our funding sources at the NIH, as well as the T32 that I am a part of. And here are my references.